All right, welcome, ladies and gentlemen, to our lesson on the immune system. So, this is going to cover the three topics we did in class, which were the introduction to the immune system, the innate immune system, and the adaptive immune system. So, what is the function of your immune system? Well, in a nutshell, it's to protect your body from pathogens. And a pathogen is any organism that causes disease in your body. So these can be, among other things, viruses, like what you see here. That is a diagram of the structure of the coronavirus that causes COVID-19. They can be bacteria, like what you see here. These are, I believe, Staphylococcus aureus bacteria, which can cause staph infections. They can be fungi. This is a close-up of the fungus that causes athlete's foot. So if you have ever had athlete's foot and you were able to look at it under a microscope, that might be what you'd see. You can also have infectious or parasitic worms that can infect your digestive system. Then you can also have other organisms. So, for example, in this central picture, what you see is parasitic amoebas. And these are amoebas that can infect you and cause serious disease and or death. In fact, we do have some parasitic amoebas that live in creeks and small bodies of water here in Texas, which is why you're not supposed to go swimming in them. So what structures are involved in the immune system? Well, there are three major organs that we're going to focus on. The first one is the bone marrow. This is the site of hematopoiesis, which is, I know, a complicated word, but it means formation of new blood cells. That prefix heme means blood. So just keep that in mind because we'll see other words that will start with heme and they will refer to blood. Hematopoiesis is the process of making new blood cells, and this includes both red blood cells and white blood cells. But for the purposes of the immune system, we're going to focus on the white blood cells that are made. Now, once cells are produced in the bone marrow, some of them may stay there to mature, and these are the ones we know as B cells, and others will actually move to another organ called the thymus to mature. So the thymus is located just above your heart, kind of a little behind your lungs. So just posterior to the lungs, but superior to the heart. It's a gland that's kind of small, but it's important because it's the site where your T cells mature and they learn not to attack your body. Another important immune system organ is the spleen. Its job is not to produce immune cells, but its job is to kind of clear things out from the blood. So down here you see the spleen. It is lateral to the stomach, so to the side of the stomach. And its job is to filter the blood. It can take out a number of things, microorganisms. It also has an important role in removing damaged or old erythrocytes, which are, by the way, red blood cells. And so you can actually live without a spleen. However, your body will not be as efficient in some of these processes. You can also have some immune cells that are going to live in the spleen or stay in the spleen for a while. Apologies for the runny nose. There are two major tissues that are involved in the immune system. They're both the fluid connective tissues, so blood, which is what is going to circulate to all parts of the cell to distribute leukocytes. And if you don't remember what leukocytes are, these are white blood cells. So here you see a number of different types. Here is a white blood cell, here is a white blood cell, and here is a white blood cell. Those are three different types. We're not going to learn about each of those. And then you also see the red blood cells or erythrocytes circulating in the blood. Then in the lymph, this is basically your liquid in your body goes from blood to what's called interstitial fluid, which is the fluid that surrounds your cells. And then it has to drain somewhere so that way it can be returned to your blood. So it flows from there to your lymph. So here you have all the liquid that's been surrounding your cells flows to your lymphatic vessels. Those can then go through your lymph nodes. You can collect leukocytes there or white blood cells, specifically lymphocytes. That's kind of a hot spot of where they'll hang out. And then also fluids will collect there and eventually drain into the blood. So it's like the blood and the lymph kind of form this kind of circle that's connected by interstitial fluid. So basically, this connects the circle, going from lymph back to the blood, which then is going to leak fluid out into the interstitial fluid, which is then going to get collected by the lymph and returned back to the blood. So it forms this nice circulation pattern. And these are lymphocytes. Those are, as well as macrophages, a couple of types of immune cells, and these are, I believe, in a lymph node so that you can see the kind of lymphatic tissue. There are a number of different cells that are important. We're going to focus on a couple of categories. These are the phagocytes. Phago literally means to eat. Cyte means cell. They are not eating cells, they are cells that eat. So here we see the process of phagocytosis from an immune cell. It is taking in these bacteria. It is basically going to swallow them and form a little ball surrounding that bacteria inside where it can break it down and digest it. This can also happen some to some damaged cells and maybe a little to infected cells. We're going to just stay with that story because otherwise we're going to start getting into some really crazy different types of leukocytes. So there are also lymphocytes, which are collectively the B and T cells, and these are part of the adaptive immune system and they will target specific pathogens. By the way, what we're seeing down here is this is the process of hematopoiesis. And forgive me, I always spell hematopoiesis wrong. I'm pretty sure that's wrong. But anyway, that is the process of the formation of blood cells, and you'll see multiple types come out. Red blood cells, platelets, and various types of white blood cells as well. So how do we divide up the immune system? Well, we can do it a number of ways, but the most common way is by function. The innate immune system is non-specific. What that means is it's going to protect against everything. And because it's kind of general all purpose, it is ready to go immediately. So within minutes, if not, at most an hour of your infection, the innate immune system is working. It is constantly active. And some of your basic mechanisms are fever, raising your body temperature, inflammation, which includes swelling and redness, barrier defenses. There are chemical signals that work in the innate immune system, and then there are phagocytes, in this case, natural killer cells. You only need to know the name, because I thought the name would be cool. Then, the adaptive immune system, though, is specific. It targets a particular pathogen, and because it has to be tailored to that pathogen, it has a slow response. It is not active all the time, it is activated by the infection. And while the innate immune system can act in the range of minutes, the adaptive immune system can take days. How does the adaptive immune system work? Well, a large part of that immunity is dependent on antibodies and other chemical signals. There are some phagocytes involved, cytotoxic T cells, and there are memory B and T cells as well, which we'll talk about towards the end. So this is just kind of showing you innate immunity versus adaptive immunity. We'll talk about each of the types of barriers. We'll not talk in too much detail about the cellular defenses. And then for adaptive immunity, you can talk about active or passive immunity. Active immunity is basically says that you're making your own T and B cells. Passive immunity means you're just getting antibodies, but you cannot make those antibodies yourself. So passive immunity only lasts until those antibodies get broken down, and no protein lives forever. So it's going to happen. And there are both natural methods of developing active immunity and natural methods of developing passive immunity as well as artificial means. So, for example, the artificial way of inducing active immunity is through vaccination. It pre exposes your immune system to some of the molecules that will be on the surface of a pathogen and will let your immune system start to develop that adaptive immune response. Or it can be artificial in that you are given antibodies from somebody who has already gotten this infection and has survived it. The passive version of that is basically through breast milk. So when you have very young babies that are still breastfeeding, they will get some antibodies from their mother, which is part of why it's considered in general, when possible, healthier to give babies breast milk to feed them on that rather than formula, just because formula does not contain these antibodies to give passive immunity. And then for adaptive immunity, the natural version is you catch a virus or a bacterium, you catch the disease. So let's talk about these two divisions. The innate immune system, the first line of defense is barrier defenses. And like I said earlier, they're constantly active and they have a couple of jobs. They can prevent pathogens from entering the body, they can destroy them right after they enter, or they can work on flushing them out before they can start establishing a population and becoming more difficult to remove. There are three types of barrier defenses: physical, chemical, and biological barriers. And we're going to talk about each of them one by one. This is just a list of various barrier defenses. Skin can be both a physical barrier as well as a chemical barrier. There are chemical barriers in multiple parts of your digestive system. Usually it has to do with either enzymes destroying stuff or pH, because those are going to break down or make it hard for bacteria or viruses to function. Then another physical barrier are mucosal surfaces. They're basically covered in mucus, and that mucus traps things, and then you can hopefully flush it out. There's also a biological barrier of non-pathogenic bacteria. These are bacteria that constantly live on various surfaces. So, for example, there can be some living inside your intestines. We know about those beneficial bacteria. There are also non-pathogenic bacteria living on your skin that help outcompete any sort of pathogens. There's also a physical barrier in the blood-brain barrier, which is an extra barrier inside your body to keep your blood from directly interacting with your brain cells. Instead, any blood or any materials from blood has to pass through this layer of border cells that are going to prevent the majority of the substances in the blood from escaping out into the brain. Your brain is so precious, your body's like, nah, don't mess with the brain. So the types of barrier defenses are mechanical barriers like this one, and they're fairly easy to note. They're the physical barriers to try and keep pathogens from entering. So skin is covered with a layer of this dead keratinized epithelium. Keratin is a hard protein. It is the protein that makes up your nails, that makes up a dog or cat's claws, makes up hooves, also makes up your hair. And then, since that layer of cells is dead, there's not really any circulating blood vessels or moisture or anything. It's very dry, so bacteria have trouble surviving there. Then, even if bacteria manage to start to work their way through your upper layer of those dead keratinized skin cells, those are going to be shed. You shed skin all the time. You may see it when you see like bits of white skin that you start to pick off, but you're also shedding kind of unseen quantities of skin cells as well. Then inside the body, anything that is open to the outside environment. So let's say we have, I don't know if that's an Adam's apple or a chin. You have this outside surface, but you also have an inside surface. Let's see, that's supposed to be your sinuses in your mouth. And then here's your esophagus, and there's your trachea. And so for your esophagus, that's connected down to your stomach, that's connected down to your digestive tract, and that is a major point where you can have pathogens enter. Because you can eat something, it may have some bacteria on it, it may have started going a little moldy, but you can't see it yet. And so you need some way to fight off those pathogens so they can't establish a foothold in you and start using up your nutrients and making you sick. So you have a number of mucous membranes that are inside of your body openings. For example, your respiratory tract, your lungs, your trachea, all of those are lined with mucous membranes. The gastrointestinal tract, so this is basically everything from your mouth down to your anus, but we're going to define it as like your stomach and your intestines. The urinary tract, so this includes like the ureter and the urethra. The important one is the urethra because that's the opening that urine goes to exit the body. And then, as well as the reproductive tracts, that's not so much a concern in males because their major reproductive tract is the urethra. So it's a common opening for both sperm and for urine, and the urine makes conditions there hostile. The reproductive tract, where you're probably going to have this biggest impact of mucous membranes, is going to be the vaginal opening for females. And then these cells are most often covered with cilia, which are tiny hairs, and they basically sweep back and forth. And what they'll do is they'll sweep this mucus layer that's covering them along until it can be removed from the body. So the most common place to think about it is in your lungs. So your lung tissue, basically, this part is gas exchange. This part, though, is lined with the cilia. And what ends up happening is any pathogens that manage to get in are going to be trapped in your mucus. So let's say there's a little virus here. And it's going to get trapped in the mucus and hopefully not be able to penetrate it to get to your epithelial cells. And then those cilia are going to sweep it along. It gets swept up your trachea, which is your windpipe. And as it goes up, it's basically going to get swallowed down the esophagus. And when it gets swallowed down the esophagus, it goes into your stomach, where your stomach acid can just destroy everything. You've got a few other things, so you can sneeze or cough to get pathogens out of your nose and throat. Your tears will actually wash pathogens from your eyes. And like I said, urine makes the urinary tract, the urethra and everything, a hostile environment for pathogens, in part because it can physically like flush them past when you've got a stream of urine passing through the small tube that is the urethra. You also have chemical barriers in all of these places. They're going to destroy pathogens chemically. Very common way to do that is to modify the pH or have enzymes. So sweat, mucus, tears, and saliva can either change the pH, contain enzymes that will break down pathogens, can contain substances that are toxic to pathogens, but not to your own cells, or they can do some combination of them. Some examples, as far as pH goes, urine is too acidic for most pathogens to grow. So every time you urinate, you're making the urethra a very hostile environment to pathogens, makes it harder for them to grow. Similarly, stomach acid is also going to have a pH that is not very friendly to pathogens, so it is more likely to destroy them. Then semen contains zinc, and zinc will also kill most pathogens. You have a number of chemical defenses that your body can employ anywhere there's a possible entry point for a pathogen. Then the biological barriers are the various non-pathogenic bacteria that live on your body or in your body. So the major places where you'll find them are outside on your skin and inside on your gastrointestinal tract, or basically your stomach and your intestines. So you'll have populations of bacteria living in mainly your intestines, and these guys take up space and they also use up any food that's available. So that way they outcompete any harmful bacteria that try to grow and make it more difficult for them to. You may see things like commercials for like for yogurt and stuff with probiotics. What they're basically trying to say is that, oh, this is supposed to contain beneficial bacteria to help repopulate your gut, and this way your friendly bacteria will have a large population and help to destroy any non-pathogenic bacteria or keep them from establishing themselves. And there is a lot of growing evidence that your microbiome inside of your intestines can have a huge impact on your health in general. For example, this link here from where I got the picture says there's a possible link between gut bacteria and depression. And I'm not saying depression is caused by gut bacteria. I'm just saying they could affect it. Then, innate immunity. Oh, still innate immunity. A number of different things that happen with innate immunity. You can have a fever, when your body temperature is higher, that is more difficult for bacteria and viruses to survive than your own body cells. So while it makes your body cells uncomfortable, it won't kill them. But there's a chance it will kill or destroy bacteria and viruses. And that gives your body an extra chance of helping to fight off the infection. So your hypothalamus is a region in your brain. It's involved in homeostasis a lot. And as part of that, it controls your body temperature. Then, when your immune system starts releasing some signals that say, hey, we've got an infection, your hypothalamus will up your body temperature, just like when you up the temperature on a thermostat. And so instead of keeping your body temperature at like a nice 98.6, 98.7, it'll get higher and higher. Generally, when you're at a fever of 100.4 or higher, you are considered to have, when you're at a temperature of 100.4 or higher, you are considered to have a fever. However, if you get up to 103, 104, that's when things start getting dangerous. You need to talk to your doctor. And if you have a sustained high fever, that's when you need to go to the hospital because too much fever or too high a fever will start damaging your own cells, especially those fragile cells in your brain. And it can, especially in young children, a high enough fever can start causing seizures. Inflammation happens after injury usually, and um, we're not going to talk about all of inflammation because we talked about it in the histology unit, but just a few things I wanted to emphasize. So, there is a chemical that's important in this inflammation. It's called histamine. I bring it up mostly because it's also involved in allergies, and we'll talk about those later. Its major action is it's going to cause vasodilation, which is increasing the diameter of blood vessels, so it increases blood flow. And then it also increases the permeability of capillaries and other blood vessels, which means it makes them leaky. So that way, liquids can very easily leak out of your blood vessels and they can take white blood cells with them. This increased blood flow will help immune cells get to the site of injury, but it will also help take up traces of what are called antigen, which we'll talk about in a little bit, to your lymph nodes. So that way it can help and develop the adaptive immune response. Another thing that will happen is that phagocytes will be drawn to the area to start eating up debris and any infectious things that are in the area. There are a number of chemical signals. There are a couple of different cell types. So we talked about the phagocytes. There are some others that trigger what we call apoptosis, and that is programmed cell death. That is basically sacrifice one to save all. So sacrifice an infected cell so that way the infection doesn't spread to save all cells, meaning the body. There are also chemical signals that work. You don't need to know the name of these. I just put them there so if you see them in a question, you'll recognize them. They're called cytokines and chemokines. They have slightly different functions, but in general, they're chemical signals that tell neighboring cells, hey, step up your game. There's an infection in the area. And they will also help call immune cells to the site of infection. There's a particular class of these chemicals called interferons, and they are specifically designed to respond to viruses, which you may hear about when you're reading or hearing about various COVID interactions with the immune system. Now, adaptive immunity or the adaptive immune system. It all depends on these two molecules. An antigen is an antibody generating molecule. It is basically anything that your immune cells can react to. So you have something on the outer surface of a pathogen, and this is a super simplistic shape, but you will have various molecules on the surface of a pathogen. Well, anything that your immune system can react to and mount a defense against or create an antibody against is called an antigen. So this guy right here has two antigens, that circle and that triangle, two potential places where your immune system can react to it. And it makes sense to have the outside of the pathogen being what triggers the immune system because all of this stuff on the inside, well, that stuff is not exposed to the outer world, which means your immune system can't see it easily. An antibody is a molecule produced by the immune system that bonds very specifically to an antigen. It's also a Y-shaped molecule, so in some diagrams, you'll see a Y shape coming out of cells. Those are actually antibodies. And depending on how many antigens are on the surface, you can actually have a single pathogen have multiple antibodies that react to it, which is a good thing. So let's talk. Here on this slide, what you're seeing is you see antibodies. Notice there are these Y-shaped purple things, and they are reacting to these antigens. So notice how they're binding to these antigens. And we're going to talk about why that's significant later, but another thing is notice how they're kind of making these pathogens, whatever they are, clump up. That's another function of antibodies. Now, if we look at this bacterial cell on the right, you'll notice it's got different antigens on the surface. These cube ones, these cone ones, and these kind of dome-shaped ones. Notice that we have antibodies that react to differently shaped antigens. But let's say this bacterium developed a mutation, and it was all of a sudden able to change the shape of those domes, so that way the antibody no longer binds. Well, luckily, your body has an antibody that reacts to another antigen on the surface, so you won't go completely blind to that bacterium. Your immune system will still be able to detect it. So that's an important feature of having multiple antibodies react to a single pathogen. It's not something that your body can necessarily say, "Oh, hey, let's be extra sure. Let's make an antibody to another pathogen." But if you have a pathogen that multiple antibodies can react to, that's usually a good thing. So, how do you identify antigens? Well, you've got these specific phagocytes that will wander around and they will eat some bacteria or whatever pathogen they see. They're going to take it in, they're going to start digesting it, which is what happens here. Then they're going to take bits and pieces of the outside, put them on some proteins, and then just stick them on the surface. So it's like basically a sign saying, I don't have enough room to write. So those are eyeglasses. It's saying be on the lookout for this guy. And then that can be presented, is what it's called, to immune cells and can train them or can determine if those immune cells will be able to react to this guy and therefore be able to start attacking it with antibodies and such. You don't need to know about which specific cells do this. You don't need to know a lot of the details on this diagram. Mostly, I like the pictures. This is another image of that. So here you see a phagocyte eating a bacterium right here. This nasty little guy who's got that little red pimple looking thing on his surface. The bacterium is digested inside of this phagocyte. It's going to take basically part of the outer part of the corpse of that bacterium, stick it on its surface, which can then go on to activate a helper T cell. That helper T cell, once it's activated, then goes on to turn on the rest of the adaptive immune system. So these guys, helper T cells, are very important. Unfortunately, HIV likes to take these out. So that's why HIV is so dangerous, because it basically kills the cells that turn on your entire adaptive immune system. So it's very interesting, but your body can actually make a bunch of different B cells and T cells with different antibodies. How do you know which one is going to react with whatever pathogen? Well, when the pathogen comes in, your lymphocytes or your leukocytes are going to be like, hey, let's take pieces of this to the place where the B cells and the T cells hang out. Hey, any of y'all got an antibody that's going to match this antigen that we have here? So you see all of these different shaped antibodies on the surface. And now you see that this antigen that's been brought in matches or seems to bind to this particular antibody. And so that guy, once he gets an antibody that binds to him, is going to be able to start reproducing and is going to start dividing. And once it does that, it's going to be able to produce a bunch of antibodies and do its job, basically. Well, as far as antibodies go, your body can make an astounding number of antibodies from just like 400 or so genes. That's an approximation, not an exact number. From this, it's thought that your immune cells can create somewhere up to 1 quintillion different antibodies. That is a 1 with 18 zeros behind it, or a billion trillion. That's a lot of antibodies. Notice these antibodies are highly specific to their antigen, so they bind to an antigen with a specific shape. So if I stuck this antibody in a pool with those 6 antigens, it would only bind to that yellow antigen. That's the only one. Now, you have to make sure, though, that your B and T cells do not react to your own cells, because if they start attacking your own cells, that's what causes what we call an autoimmune disorder. If it's autoimmune, that literally means self-immune. Doesn't mean you're immune to yourself. It means your immune system attacks yourself. And this is really evident when you look at the ABO blood group or blood typing system. So there is a specific antigen on the surface of red blood cells. It can be either this A shape, which is a circle, this diamond shape, which is representing a B. You can have actually both of those antigens on the surface, or you can have none of them. That's what gives you your blood type of A, B, AB, or O. Because of that, if you have type A blood, your body actually makes anti-B antibodies. So if you end up taking in blood cells that have the B antibody, your body is going to attack them and is going to destroy them, which is why a type A person cannot be given type B blood. Conversely, with type B blood, those cells of those body's going to make anti-A antibodies. So if it comes across something with an A antigen, then it is going to attack it, and then it is going to basically destroy those cells. Then your type AB blood cells, the person with that blood type doesn't make any antibodies, because if they did, they would be attacking themselves. Type O blood types, they make antibodies against type A and type B, which means they can't receive anything with an A or a B on it. So they can only get blood from other type O people. And this is called learning to recognize self versus non-self. Your self antigens are the one from your own body. So your self antigens are these guys. And your non-self antigens are basically everything else. So in a type A person, their body is going to produce cells with that A antigen, and they're going to make anti-B antibodies. So if you give them type B or type AB blood, they are going to get sick. The new blood cells are going to be destroyed. It's going to start causing clogs in their blood vessels, and they could get very sick and die. So you have to train these cells so that they learn not to attack self-antigens. So when you have T cells, say, for example, in the thymus, they're going to be presented with a self-antigen by a cell in the thymus. Not important to know what it is. The important thing to know is that if it binds very heavily to that T cell is going to die. That way, you don't produce or keep around a T cell that's going to attack your own cells. On the other hand, if the T cell doesn't react very strongly to an antigen, so this is that same T cell, but it's reacting very weakly to another self-antigen, since it doesn't really react strongly to it, it gets to live. And if it gets to live, what it gets to do is go out, wander around, and if it encounters a cell, this is a virally infected cell, and it encounters that antigen, it will destroy that cell. So you want this cell, whatever it is, to not be one of your body cells, because if you start destroying your body cells, well, how are you going to be able to stay alive? So what do T cells do? Their main job is to activate other immune cells. So B cells and other parts of the adaptive immune system will not function, will not get activated without T cells. They have a number of other functions as well that are listed here. And an important thing that they do is they turn off the immune response once the pathogen is gone. That's very important because without that, your immune system would just keep going. And if it didn't have a target to attack, it might attack your cells. B cells, their major role is they make antibodies. And there are also types of B cells called memory B cells that can stick around in your body for decades. That way, the next time you get attacked by that same pathogen, your body doesn't have to pause and find an antibody that will match it. And these guys are trained just like T cells are so that they will not attack body cells. So B cells are activated by T cells. So let's say you have a B cell, it runs across a bacterium with whom it has, it matches the antigen basically, they fit. Well, that will cause that B cell to be like, okay, I think I got something. If it is in turn activated by a helper T cell, like the helper T cell you can think of as kind of double checking its work, then if it's activated, it can divide and make a bunch of what are called plasma cells and some memory cells. The memory cells we'll talk about in a moment. The plasma cells, their job is to make antibodies. The reason those antibodies are significant is because they signal the rest of the immune system, hey, there's a pathogen there. Let's eat it. And so phagocytes will come and they will eat those antibody covered pathogens. Then later, if you get infected by the same pathogen, you still have memory cells around, and what they're going to do is they're going to immediately be able to start turning on the rest of your immune system and getting your immune system, your adaptive immune system, up and running very quickly to attack that pathogen and get rid of it so quickly you may not even know that you are sick or infected. And that is it. I will see you next time for our next lesson.